Sensation and perception. Most people experience an integrated percept of the world. However, that isn't always the case for people who have suffered localized brain damage. For example, LM's visual experience of the world was markedly changed following a stroke. Viewing moving objects made her feel quite unwell. She reported that she no longer saw motion, rather that objects in motion now seemed restless or jumping around. She could still correctly perceive static objects, but not their motion when they moved. LM's perception of motion appears to be like watching a movie sequence, but seeing only a few frames in the sequence, with each frame persisting until the next is perceived. She sees the objects frozen in one location, and then they jump to a new location. As a consequence, activities like crossing a busy road are dangerous for her because cars that appear distant and stationary suddenly jump to locations near her. Even pouring water into a glass is difficult. The water appears frozen as if ice, and then appears at a new level, and then it overflows the glass. On the other hand, Dr. P, who suffered damage to a different region of the brain, had no trouble perceiving objects and their motion. However, by merely looking at them, he could not identify what they were. He could fully describe the object, but had no idea as to its identity, to the extent of having even mistaken his wife's head for a hat. There's a book on that. These two cases give us some insight into how visual information is processed by the brain. LM suffered damage to the lateral temporal occipital region of her cortex, which is part of the dorsal pathway, while Dr. P suffered damage to the posterior occipital region, which is part of the ventral pathway. These specific patterns of visual impairment due to localized brain damage clearly indicate that there is localization of function in the brain. That is, cells in particular locations are specialized to extract specific types of information from the visual scene. However, this mapping of function onto the brain is particularly complex and can only be fully understood by knowing how the visual system breaks the scene down and reconstructs that information in order to create a complete percept of the world. For example, it is a mistake to think that a particular cortical region processes a specific attribute like motion or object identity. This is due to two defining aspects of how the brain processes visual information: parallel pathways and multiple processing stages. The patterns of deficits following localized damage to the brain are complex and cannot be understood without knowing how the brain processes sensory information. While great advances have been made in our understanding of this process, more research needs to be done. However, by the end of this chapter, you will understand both the fundamental principles of how the brain generates our percept of the outside world, and also why particular patterns of deficits following localized brain damage occur. The visual system doesn't consist of just a single pathway, but rather multiple pathways that are specialized to extract different aspects of the scene. Cells in the dorsal pathway have been linked to processing information about where an object is, in both space and time and motion information, while ventral cells have been linked to determining what the object is. In relation to multiple processing stages, there are two aspects of this concept that are relevant here. The first is that all stimulus features are processed in multiple stages along these parallel pathways, with the information being transformed at each stage, for example, colour and motion. So there's not a single location within, for example, the dorsal pathway that processes motion. Additionally, a given stimulus feature can be used for many different purposes. Motion information can tell us about the change in location of an object, that is, its actual motion, which is processed by the dorsal pathway. However, motion signals can also be used for other purposes, for example, communicating form information, specifically the three-dimensional structure of an object, and hence processed by the ventral system. There is no single region in the brain that fully processes a particular attribute. Therefore, a person will never be totally blind to a single aspect of perception, like motion or colour, due to localized damage to brain. For example, motion information is processed at different stages within both the dorsal pathway to extract different aspects of the motion of objects and also at different stages in the ventral pathway to extract form information. It is only by knowing how cells at various stages in these pathways break down the visual scene and how that information is recombined by interactions between those different stages and pathways that one can fully understand the various patterns of deficits and retained abilities following localised brain damage. The fact that these specialised cortical areas process different aspects of our perceptual world, over 25 distinct areas have been identified just for visual processing, results in the binding problem, which is one of the big mysteries of cognitive neuroscience. How do we bind together all those distinct sensory components into one complete perceptual whole? When you hold a rose in your hand, see its coloured petals, feel their velvety quality, and smell its aroma. These disparate sensory experiences are somehow fused into your total perceptual experience of the rose. In some ways, sensation and perception blend together so completely that they are difficult to separate, for the stimulation we receive through our sense organs is instantaneously organised and transformed into the experiences that we refer to as perceptions. Nevertheless, psychologists do distinguish between them. Sensation is the stimulus detection process by which our sense organs respond to and translate environmental stimuli into nerve impulses that are sent to the brain. Perception, making sense of what our senses tell us, is the active process of organising the stimulus input and giving it meaning. Because perception is an active and creative process, the same sensory input may be perceived in different ways at different times. There's an example on page 147 where it asks you to read the two sets of symbols in figure 5.2. Pause the recording and have a look, and then come back to this. Okay, so the middle symbols in both sets are exactly the same, and they sent an identical input to your brain, but you probably perceived them differently. Your interpretation or perception of the characters was influenced by their context, that is, by the characters that preceded and followed them, and by your learned expectation of what normally follows the letter A and the number 12. This is a simple illustration of how perception takes us beyond sensation. Figure 5.1, sensation becomes perception. The stage involved in perceiving an object. The object projects an image onto the eyes, thus creating a retinal image. It's the information, proximal stimulus, that the brain has to use to generate a percept of the object, distal stimulus. Visual illusions, misperceiving the word, can occur due to errors in the sensory encoding of information, sensory illusions, or in how the brain interprets the sensory information, perceptual illusion. So the object is the distal stimulus, then it goes to image projection, then proximal stimulus, which is the retinal image, then the sensory representation of proximal stimulus, and then the perceptual representation of distal stimulus, which is the perceived object. Sensory processes. The particular stimuli to which different animals are sensitive vary considerably. The sensory equipment of any species is an adaptation to the environment in which it lives. Many species have senses that humans lack altogether. Carrier pigeons, for example, use the Earth's magnetic field to find their destination on cloudy nights when they can't navigate by the stars. Sharks sense electric currents leaking through the skins of fish hiding in undersea crevices, and rattlesnakes find their prey by detecting infrared radiation given off by small rodents. Whatever the source of stimulation, its energy must be converted into nerve impulses, the only language the nervous system understands. The key term here is transduction, and transduction is the process whereby the characteristics of a stimulus are converted into nerve impulses. We now consider the range of stimuli to which humans and other mammals are attuned and the manner in which the various sense organs carry out the transduction process. As a starting point, we might ask how many senses there are in humans. Certainly there appear to be more than the 5 classical senses: vision, audition, gustation, olfaction, and touch. For example, there are senses that provide information about balance and body position. Also, the sense of touch can be subdivided into separate senses of pressure, pain, and temperature. Receptors deep within the brain monitor the chemical composition of our blood. The immune system also has sensory functions that allow it to detect foreign invaders and to receive stimulation from the brain. Like those of other organisms, human sensory systems are designed to extract from the environment the information that we need to function and survive. Although our survival does not depend on having eyes like eagles or owls, noses like bloodhounds, or ears as sensitive as those of the worm-hunting magpie, we do have specialised senses that can detect many different kinds of stimuli with considerable sensitivity. The scientific area of psychophysics, and that's a key term, which studies relations between the physical characteristics of stimuli and sensory capabilities, is concerned with two kinds of sensitivity. The first concerns the absolute limits of sensitivity. For example, what is the dimmest light, the faintest sound, or the weakest salt solution that humans can detect? The second kind of sensitivity has to do with differences between stimuli. What is the smallest difference between two tones that we can detect? Stimulus detection, the absolute threshold. How intense must a stimulus be before we can detect its presence? Researchers answer this question by systematically presenting stimuli of varying intensities to people and asking whether they can detect them. Researchers designate the absolute threshold as the lowest intensity at which a stimulus can be detected. That's a key term, the absolute threshold. Thus, the lower the absolute threshold, the greater the sensitivity. Another key term is subliminal stimuli, and it says that note that subliminal stimuli, which is very weak stimuli that do not register in awareness, can also influence behaviour. From studies of absolute thresholds, we can estimate that general limits of human sensitivity for the 5 major senses. Some examples are presented in Table 5.1, and this table shows the sensory modality and the absolute threshold. So the first sensory modality is vision. The absolute threshold is candle flame can be seen at 50 kilometres on a clear dark night. The next sensory modality is hearing. The absolute threshold is the tick of a watch under quiet conditions at 6 metres. Taste, 1 teaspoon of sugar in 7.5 litres of water. Smell, 1 drop of perfume diffused into the entire volume of a large apartment. And touch, wing of a fly or bee falling on a person's cheek from a distance of 1 centimetre. Many of our absolute thresholds are surprisingly low, yet some species have sensitivities that far surpass those of humans. For example, a female silkworm moth that is ready to mate needs to release only a billionth of a gram of an attractant chemical molecule per second to attract every male silkworm moth within a 1.6k radius. At one time, scientists thought that although some people have greater sensory acuity than others, each person has a more or less fixed level of sensitivity for each sense. But psychologists who study stimulus detection found that an individual's apparent sensitivity can fluctuate quite a bit. The concept of a fixed absolute threshold is inaccurate because there is no single point on the intensity scale that separates non-detection from detection of a stimulus. There is instead a range of uncertainty, and people set their own decision criterion. Decision criterion is also a key term, and that is a standard of how certain they must be that a stimulus is present before they will say they detect it. The decision criterion can also change from time to time, depending on such factors as fatigue, expectation, such as having watched a horror movie, and the potential significance of the stimulus. Signal detection theory, another key term, is concerned with the factors that influence sensory judgments. So this next bit is also a figure, which is 5.3 on page 148. They've changed the way they show the page numbers now. So have a look at that, and I'll read. In a typical signal detection experiment, participants are told that after a warning light appears, a barely perceptible tone may or may not be presented. Their task is to tell the experimenter whether or not they hear the tone. Under these conditions, there are 4 possible outcomes, and these are shown in figure 5.3. When the tone is in fact presented, the participant may say yes, which is a hit, or no, which is a miss, and when no tone is presented, the participant may also say yes, which is a false alarm, or no, which is a correct rejection. At low stimulus intensities, both the participants and the situation's characteristics influence the decision criterion. Bold participants who frequently say yes have more hits, but they also have more false alarms than do conservative participants. Researchers can influence participants to become bolder or more conservative by manipulating the rewards and costs for giving correct or incorrect responses. Increasing the rewards for hits or the costs for misses results in lower detection thresholds, so more yes responses at low intensities. Thus, a Navy radar operator may be more likely to notice a faint blip on the screen during a wartime mission, when a miss could have disastrous consequences, than during a peacetime voyage. Conversely, like doctors who will not perform a risky medical procedure without strong evidence to support their diagnosis, participants become more conservative in their yes responses as costs for false alarms are increased, resulting in higher detection thresholds. Signal detection research shows us that perception is, in part, a decision. Subliminal stimuli, very weak stimuli that do not register in awareness, can also influence behaviour. The difference threshold. Distinguishing between stimuli can sometimes be as important as detecting stimuli in the first place. When we try to match the colours of paints or clothing, small stimulus differences can be very important. Likewise, a slight variation in taste might signal that food is tainted or spoiled. Professional wine tasters and piano tuners make their living by being able to make subtle discriminations. The difference threshold, which is a key term, is defined as the smallest difference between two stimuli that people can perceive 50% of the time. The difference threshold is sometimes called the just noticeable difference, or JND. German physiologist Ernst Weber discovered in the 1830s that there is some degrees of lawfulness in the range of sensitivities within our sensory systems. Weber's law, which is also a key term, states that the difference threshold, or JND, is directly proportional to the magnitude of the stimulus with which the comparison is being made, and can be expressed as a Weber fraction. For example, the JND value for weights is a Weber fraction of approximately 1/50th. This means that if you lift a weight of 50 grams, a comparison weight must be at least 51 grams in order for you to be able to judge it as heavier. If the weight were 500 grams, a second weight would have to be at least 510 grams, i.e. 1/50th equals 10 grams of 500 grams, for you to discriminate between them. Parallel pathways in the brain. Two defining aspects of how the brain processes visual information are parallel pathways and multiple processing stages. The visual system consists of many distinct pathways, with cells in those pathways having specific tuning properties. That is, they are sensitive to particular aspects of the visual world. This, in turn, means that these pathways are extracting specific types of information about the visual scene. Parallel pathways occur within the visual system from the retina onwards, and they can be identified at many different scales. At the cortical level, the two main parallel pathways are the ventral and dorsal pathways. These are specialised for the extraction of form and spatial information, location and motion information, respectively. And they, in turn, can be broken down into distinct parallel pathways. For example, the dorsal pathway can be divided into pathways specialised for extracting spatial location and motion information, and the motion system itself can be further subdivided into pathways extracting different types of motion information. Each of these pathways contains a number of distinct processing stages, and the information presented in the pathways is transformed as it moves from one stage to the next. Given these aspects of how the visual system works, a fundamental question that could be asked is, to what extent and at what levels do these pathways interact with one another? Initially, it was thought that they were largely independent. However, recent research has discovered that there is substantial crosstalk between them. The research has focused on how motion and form signals interact. Initially, it was thought that motion information was processed exclusively in the dorsal pathway and form information in the ventral pathway, and so they did not interact with one another. However, research has shown that there is substantial interaction between them. An early example of form-motion interactions is the demonstration of biological motion, in which the complex patterns of motion that a person's joints make as they walk, also called a point light walker, can be easily interpreted by people to determine that it is a person walking, dancing, or the like. A number of studies, including many by Australian researchers, have focused on what are called motion streaks, which are thought to be a way for form information to enhance the ability of the visual system to extract motion information. Such facilitation would be of particular benefit in situations where the visual scene is very complex and cluttered, making it difficult to extract the motion of moving objects from all the random motion. Motion streaks result from the response persistence of cortical neurons. That is, cortical neurons continue responding for a brief time after the stimulus that elicited the response has moved on, resulting in a smeared representation along the direction of the motion in the brain of the moving stimulus. And figure 5.4 shows two images of a creek with running water. One is how we see it, or how we think we see it, and the other one is the same image but taken with the camera using a slow shutter speed. This constitutes a form cue that is not present in the visual world itself. We typically aren't aware of these motion streaks, that is, we don't typically perceive them, though one condition under which we do perceive them is when watching fireworks displays. There, probably due to the high contrast of the bright fireworks against the black sky and the retina being in a dark adapted state, we see the moving dots of light that make up the fireworks as being streaks of light. Motion streaks can then be detected by form-sensitive neurons, possibly standard orientation-selective simple neurons on V1. These neurons can signal the object's axis of motion, that is, both the actual and opposite direction of motion of the object. The response of these form cells is then combined with the response of actual motion cells so they can increase the response of the motion cells. Even with a noisy motion stimulus, the response of the motion cells turned to the object's direction of motion would be greater than the response of cells turned to the opposite direction. Hence, by facilitating the response of motion cells turned to both of these directions, the net result will be a stronger response in the cells signalling the correct direction of motion, and therefore a stronger and cleaner perception of motion. Research has shown quite convincingly that motion streaks play an important role in motion perception. For example, thresholds are lower for stimuli that result in longer motion streaks. Simulated motion streaks, i.e. short line elements that are briefly presented, bias the perceived direction of a moving object toward the direction defined by the orientation of the line elements, and strong motion streaks result in adaptation of form-specific cells. I actually don't understand a word of what I'm reading. Something about motion streaks, and dots. The form system is not the only pathway that provides input into the motion system. Such input also comes from the vestibular system and is used in the processing of optic flow motion signals. The vestibular system is part of the middle ear and consists of fluid-filled chambers. It responds to inertial forces produced by changes in our speed and direction of movement, and it is important for our sense of balance. Optic flow signals are the patterns of motion that the images on our eyes undergo as we move through the world, with particular patterns indicating the specific types of movement. For example, a radially expanded pattern, as used in science fiction movies such as Star Wars, when a spacecraft jumps to light speed, indicates forward motion, while a contracting pattern indicates backward motion. While optic flow signals are important in helping us move through an environment, they are also key in maintaining balance while standing. Compare standing on one foot with your eyes open to standing on one foot with your eyes closed, i.e. when you have removed the optic flow signals. Testing sensitivity to optic flow signals while also physically moving the person in either a consistent or inconsistent manner shows that people are more sensitive in the consistent condition. This finding provides strong evidence for input from the vestibular system into the motion system, and such interaction makes sense given the relative sensitivities of the two systems. While both are very sensitive to motion depth, the optic flow system is not very sensitive to variations in speed, while the vestibular system is. The implications of these various interactions are varied and important and include increasing our understanding about 1. patterns of impairments and retained abilities in clinical neuropsychology. For example, the so-called motion-blind patient LM was initially thought to be totally and selectively blind to motion. However, the first stage of motion processing, detection of motion, occurs in the primary visual area. So if that was damaged, other visual abilities would be impaired. LM has damage to the later stages in the dorsal motion system, which are involved in pooling motion signals to do a range of tasks, including the extraction of complex motion signals such as optic flow patterns. However, LM identified a point light walker, that is, she pooled motion signals to extract form information, ventral task, but was not able to pool these motion signals to determine the actual direction of motion of the walker, a dorsal task. Number 2, synesthesia, which likely results from crosstalk between stages in the various parallel pathways that shouldn't be occurring. Number 3, motion sickness. The interaction between the vestibular and optic flow system is likely to play a significant role in generating the feeling of motion sickness, which occurs when the two systems are indicating different types of motion. Although Weber's law breaks down at extremely high and low intensities of stimulation, it holds up reasonably well within the most frequently encountered range, thereby providing a useful barometer of our abilities to discern differences in the various sensory modalities. Table 5.2 lists Weber fractions for the various senses. This is found on page 151. The smaller the fraction, the greater the sensitivity to differences. As highly visual creatures, humans show greater sensitivity in their visual sense than they do in, for example, their sense of smell. Undoubtedly, many creatures that depend on their sense of smell to track their prey would show quite a different order of sensitivity. Weber fractions also show that humans are highly sensitive to differences in the pitch of sounds, but far less sensitive to loudness differences. Sensory adaptation. From a survival perspective, it's important to know when some new development requires your attention. If you were relaxing outdoors, you would want to be aware of the whine of an approaching mosquito. Because changes in our environment are usually noteworthy, sensory systems are finely attuned to changes in stimulation. Sensory neurons are engineered to respond to a constant stimulus by decreasing their activity. The diminishing sensitivity to an unchanging stimulus is called sensory adaptation. And that's a key term. Adaptation, sometimes called habituation, is part of everyday experience. After a while, monotonous background sounds are largely unheard. The feel of your wristwatch against your skin recedes from awareness. When you dive into a swimming pool, the water may feel cold at first because your body's senses respond to the change in temperature. Over time, however, you become used to the water temperature. Adaptation occurs in all sensory modalities, including vision. Indeed, were it not for the tiny involuntary eye movements that keep images moving about the retina, stationary objects would simply fade from sight if we stared at them. In an ingenious demonstration of this type of adaptation, R.M. Pritchard, 1961, attached a tiny projector to a contact lens worn by each participant, figure 5.5a, which is also on page 151. This procedure guaranteed that visual images presented through the projector would maintain a constant position of the retina even when the eye moved. When a stabilised image was projected through the lens onto the retina, participants reported that the image appeared in its entirety for a time and then began to vanish and reappear as parts of the original stimulus. That's on figure 5.5b. I did not know that about our eyes making little movements to keep stationary objects from fading. Imagine that they would fade if they didn't jump around the back of your eye. So to create a stabilised retinal image, a person wears a contact lens to which a tiny projector has been attached. Despite tiny eye movements, images are cast on the same region of the retina. Under these conditions, the stabilised image is clear at first and then begins to fade and reappear in meaningful segments as the receptors fatigue and recover. Although sensory adaptation may reduce our overall sensitivity, it is adaptive, for it frees our senses from the constant and the mundane, allowing them to pick up informative changes in the environment that could be important to our well-being or survival. There's a concept check. So sensation is how our brain receives and encodes information. Perception is how it interprets that information. An absolute threshold is the lowest intensity of a stimulus that we can reliably detect. A relative threshold is the smallest difference between two supra-threshold stimuli that we can reliably detect. And sensory systems are specifically responsive to changes in stimulation, and this is increased via adaptation, which is the reduction in the sensitivity of these mechanisms due to being present with unchanging stimulation.